Hi, I'm David, a PhD student in Ron Shamir's lab at Tel Aviv University. I'm going to be presenting our work on improving plasmid assembly in metagenomics. This is joint work with the Mizrahi lab at Ben-Gurion University. The goal of our work is to assemble plasmid genomes from metagenomic samples, and our contribution is a new tool called SCAPP, which achieves this. I'm going to begin with some background about plasmids. So plasmids are extrachromosomal DNA molecules which exist within bacterial cells. Plasmids can be replicated during cell replication along with the genome of the host bacteria, and some plasmids can also be transferred laterally across bacterial cells. Plasmids are of interest because they can encode genes which confer increased metabolic fitness to their hosts. So for example, antibiotic resistance genes. And then they also facilitate the transfer of these genes across bacterial populations, as the plasmids themselves can be transferred between individual bacteria. The algorithmic setting of our work is a data structure known as the assembly graph. So if we think of the genomes which are present in our sample, there's going to be some underlying unknown genome sequence. And what we observe is a sampling of short reads with errors from the underlying genomes. If we want to try to reconstruct the genomes, or at least large segments of them as much as possible, what is often done in many of the assembly tools is to construct a data structure known as the Brown graph. So the short reads are broken up into all of their overlapping k-mers, so those are k-long substrings of the read. And we build the Brown graph where there's a node for every such k-mer, and there's an edge between two nodes if the suffix of one k-mer matches the prefix of the adjacent k-mer. Once we have the Brown graph, we can compact unambiguous walks in the graph into a single sequence and into a single node. So for example, here we have an unambiguous path from GAC into AGA, and that can be compacted into what's called a contig, and that's shown in the assembly graph as a single node. So we construct the assembly graph by compacting all of these graphs into single nodes and looking at the underlying contigs, which can be much longer than any individual read in the sequencing itself. So metagenomic assembly is a challenging task. We're dealing with large and complex assembly graphs. The graphs themselves are tangled or fragmented due to strain variation or overlap in genetic content between different bacterial species. And when we try to assemble the plasmids, we're actually trying to assemble only a very small fraction of this metagenome. And so we're really looking for a needle in a haystack in this tangled and messy assembly graph. And the plasmids themselves share sequences among each other and with their host bacterial genomes, and so they're going to be embedded in some tangled portion of the assembly graph. Prior to our work, there were two previously existing tools. One is known as Recycler, that came out of our lab a number of years ago, and the other is a variation of the Spades assembler, which was tailored for assembling plasmids from the metagenome. So Recycler tries to identify uniformly covered cycles in the assembly graph, and in an iterative process, it removes each cycle from the graph and looks for another cycle in the updated assembly graph. MP Spades is also an iterative algorithm. First, they look for cycles in the entire assembly graph, and then they have an increasing coverage threshold where in each iteration they remove all contigs whose coverage falls below the current threshold. That could reveal longer and longer unambiguous paths within the graph, which hopefully could represent plasmids. I'm going to talk about some of the main ideas of SCAPP right now. So our input is a metagenomic sample, so the reads. We use standard tools such as MegaHit or Metaspades to construct the metagenomic assembly. We then build what we call the annotated assembly graph. So we overlay the locations of plasmid-specific genes onto the metagenomic assembly. These are from a curated database that we've constructed of known plasmid-specific genes. We also classify every single contig in the assembly with the probability that it could be from a plasmid sequence. So this is using a sequence classifier that we've trained and previously released in a PLOS computational biology paper. And we also map the metagenomic reads back to the assembly graph, and this allows us to identify paired end read links between different contigs in the graph. Once we have the annotated assembly graph, we look for uniformly covered cycles within that graph, and we're focusing only on the regions of the graph that have plasmid annotations. And if we find such a cycle, we can peel it from the graph if we believe that it's a plasmid. And then in a final step, we filter out those cycles in order to remove the false positives and retain only the true assembled plasmids. So all of the annotations in the graph are combined into a single weight, which is assigned to every node in the graph, and we want the weight to be low. So we assign the lowest weight of 0 to nodes which contain plasmid genes, and we also assign lower weights to nodes which have a higher probability of being plasmid as identified by the plasmid sequence classifier. Finally, longer or more highly covered nodes are also assigned lower weights. Once we've combined all of the annotations into a single weight for every node, we then iterate over the graph looking node by node to find the lowest weight cycle through every node. This can be done efficiently using a shortest path algorithm. So for example, if we're interested in the red node, we look at the incoming neighbors of those nodes, of that node, and we try to find the shortest path to each of those incoming neighbors, and then we decide between those shortest paths which one has the lowest weight. Once we've identified such a path, we decide whether or not it could potentially be a plasmid based on how uniform the coverage is along the cyclic path, how consistent the path is with the paired end read links between contigs on the path, and also the overall graph structure. So are we looking at an isolated loop in the graph or a cycle that was found embedded in a much more complex structure within the graph? Once we've identified such a cycle as potentially being a plasmid, we peel it from the graph. So what that means is, for example, here we have in red a potential cycle, and we have the numbers represent the coverage of those nodes. And we find the average coverage along the cycle, and we remove it from the graph. And what that does is results in a subgraph. So for example, some of the nodes, for example, this bottom one, are removed as all of their coverage has been removed from the graph, while the remaining nodes have their coverage lowered by the mean value along the cycle. More algorithmic details can be found in our paper. We're going to move on now to results. And again, we also have simulated results, which I'm not going to show here, that can be found in our paper. And I'm going to focus on results on real biological data. So the first sample that I want to talk about is a data set of human gut metagenomes. This is publicly available, and we took 20 of the largest samples from this data set. What I'm showing here are the F1 scores of the different tools. This is a combination of the precision and the recall. The score multiplied by 100 is shown here on the left. And on the right, I'm showing the number of known plasmids which appear within each sample. So that's shown with this dotted line. And as you can see, the number of samples, the number of plasmids in a sample increases along the y-axis, the x-axis. So what we see is that in many samples, SCAPP has the highest F1 score, especially in the larger samples which have the most plasmids, it has a significantly higher F1 score than existing tools. Overall, SCAPP has far higher average F1 across all of the samples, meaning that it performed the best. The second data set that I want to talk about is a human gut plasmidome that we sequenced. So this is a human gut microbiome sample which was enriched for plasmids. All of the chromosomal DNA was removed using the protocol developed in the Mizrahi lab. So in this case, we have many more known plasmids in the sample, right, over 70 compared to the 20 to 30 in the largest samples from the previous data set. And again, SCAPP has the highest F1 score. This is because both SCAPP and Recycler have higher recall than MP-Spades, while SCAPP has the highest precision overall. Combined, that allows it to have the best performance. Final data set that I want to talk about is a parallel cow-rumen metagenome plasmidome sample that we sequenced. So we first have a single cow-rumen specimen that's divided into two samples. Each of those samples is sequenced separately. One is standard metagenomic shotgun short-read sequencing. The other one is first undergoes the plasmid enrichment protocol, so all of the chromosomal DNA is removed, and then it is sequenced. And what this allows us to do is construct the assembly graph of the metagenome and then try to run each of the algorithms to find plasmids from the metagenomic sample. And then we can evaluate those plasmids based on the reads from the plasmidome. And here I'm going to show in more detail how that works. So if we have metagenomic assembly, we map plasmidome reads onto that assembly. And we identify all of the locations in the assembly graph that should be of plasmid origin. And we define the recall as the fraction of those regions which actually are assembled by each of the algorithms into plasmids. We also define a plasmidome read-based precision, where we take the final plasmids that were output by each of the algorithms, and we map the plasmidome reads directly to those plasmids, and then we look at the fraction of those plasmids which is actually covered in the plasmidome. When we do this, we see that SCAPP and MP-Spades have similar results on this data. So SCAPP has slightly higher precision and very slightly lower recall than MP spades, whereas both of those tools perform much better than recycler, which has much lower precision than either of them. Another thing that we do with this data set is we look at the union of all of the plasmids constructed by any of the tools within the plasmidome. And so this is some global ground truth set of plasmids. And we see what the performance of each of the individual tools is when constructing plasmids from the metagenome. So we see, when we do this, we see that SCAPP has the highest overall precision, and again, the highest overall F1 score. When we look at the runtime and memory of the tools, so the memory is basically the same for all of the tools because they're all doing metagenomic assembly, which is the sort of memory bottleneck of this task. And in terms of runtime, MP spades is the fastest. It does both the assembly and the identification of plasmids together in one step, whereas recycle and SCAPP both first construct the metagenomic assembly and then assemble plasmids within the assembly graph. And SCAPP is slightly slower than recycler. Overall, though, the tools are very similar to each other. In summary, we've developed a new tool called SCAPP, which is aims to assemble metagenomic plasmids based using the annotated assembly graph. We show that it improves performance in a range of biological settings while retaining similar runtime and memory usage to the existing tools. We also showed some novel experimental data and new evaluation settings. And all of our code can be found on GitHub, and the paper itself is just published in BMC Microbiome. So thank you for listening. I want to thank again my co-authors and advisor, the funders, and if you have any questions, I'm happy to take them now, or you can also email me if you want to ask anything offline. Thanks again.